In today's video, I'm going to be discussing my final lab project for BIT 480. To begin, yeast is a diverse, remarkable eukaryotic organism. A few characteristics of yeast include the fact that it can be well characterized genetically, it can be easily manipulated, it does not need a unique environment in order to grow, and it has a quick doubling time. The type of yeast I'm going to be discussing specifically is Saccharomyces cerevisiae. This strain of yeast is highly genetically tractable, is stable as either a haploid or a diploid, and has a wide variety of genetic tools available. Metabolic engineering is the intentional genetic engineering of organisms to produce useful products and substances. Metabolic engineering can be used in yeast in order to produce beta-carotene, which is a red-orange pigment found in plants and fruits. It is a hydrocarbon spanning 40 carbons with beta rings at either end of the molecule. Throughout the duration of this course, both wild-type yeast and yeast from the pooled deletion library were used in order to produce beta-carotene. This project included selecting unknown deletion strains in order to identify genetic regulators of beta-carotene production in yeast. I selected 2 colonies from the pooled deletion strain transformation for analysis based on colony phenotype, in which a single gene was deleted in each of the colonies. Before sequencing the barcodes, the regions of DNA were amplified that contain these barcodes from our yeast. The uptack and downtack barcode sequences from the colonies that I selected were then sequenced. These sequences allowed identification of the strains and determination as to which gene was deleted. This information allows us to speculate whether the genes that were deleted were negative or positive regulators of beta-carotene production, or perhaps something unrelated. I also used yeast databases and primary literature sources in order to determine if the proteins encoded by these genes were involved directly in a beta-carotene pathway. Here are my results for my 2 colonies. If a gene is deleted and this causes increased beta-carotene production, this means that a negative regulator was deleted. If a gene is deleted and this causes decreased beta-carotene production, this means that a positive regulator was deleted. Results showed that colony number 23 had a deletion of gene YGR50C. Its phenotype is completely white and is not similar to the WT-BY4741 strain, which is dark orange. I would hypothesize that this gene is a positive regulator since the colonies are white in color. The protein produced by this gene has an overall unknown function in which its molecular function, biological process, and cellular components are all unknown. Colony number 24 has a deletion of gene GLC8, which is involved in glycogen metabolism and chromosome segregation. It is also proposed to regulate GLC7P activity through conformational alteration. It is also associated with glycogen deficiency in Saccharomyces cerevisiae and increased enzyme activator activity. Because its phenotype is similar to the wild type, I hypothesize that this has no correlation with beta-carotene production. Overall, I did not find any significant connections between these genes and pathways that could influence beta-carotene production. My results led me down an entirely different pathway, with one of my gene's functions being totally unknown. If I were to follow up on a gene of interest, I would conduct further research on gene GLC8. The fact that it is associated with glycogen deficiency in Saccharomyces cerevisiae is intriguing to me.